السلام عليكم ورحمة الله وبركاته. I hope إن انتوا تكونوا سامعيني. أنا مش متأكدة فعلا إذا كان الكونكشن كويسة ولا لأ. بس يعني أنا سعيدة جدا إن أنا أبقى معاكم، دي أول مرة ليا. I hope إنها ما تكونش الأخيرة يعني. البلان بتاعي كان إن شاء الله إن أنا أقدم البرزنتيشن دي الأول. هي mainly عن genetics for breast cancer. يعني أحب أقول لكم جزء صغير أنا ليه حابة أقدم البرزنتيشن دي النهاردة. إنه الإيم بتاعي إن أنا إن شاء الله أقدمكم أو أعرفكم على البورد أوف ميديكال جينيتكس للناس اللي هم interested إن هم يجوا أمريكا ويعملوا بورد إكزام. ده واحد من السبيشاليتي اللي موجودة. إن شاء الله المرة الجاية هعرفكم على البورد، على requirement and how to apply.
دكتور أمال؟
نعم.
You hearing me?
I hear you. أنا كنت بكلم روحي.
No, we hear you from the start.
Okay.
Okay.
Continue.
Yes.
Okay. الحقيقة البرزنتيشن دي هي البرزنتيشن اللي أنا عملتها during my PhD. أنا originally كنت obstetrician in Egypt. ولما جيت أمريكا عملت PhD. الشغل ده هو اللي خلاني more interested in the genetic field. وبعد كده أخذت fellowship في genetics، وحاليا I'm board certified in genetics. So لما أبدأ البرزنتيشن تعرفوا why I was very interested إن أنا أدخل الفيلد ده. So we'll start now. The title of my presentation is 53 Binding Protein 1 Abnormality in Breast Cancer, and it was done in the Cancer Institute of New Jersey and Robert Wood Johnson Medical School. So at the beginning, I will talk about what is breast cancer. We all know breast cancer is a common disease, unfortunately, one of the major killers for women. One of the main challenges for breast cancer is it is not one disease. And there were many attempts to try to classify breast cancer. One of the classification that I introduced here, which I call it the therapeutic classification, which is depend on if there is a therapy for this subtype or not. So, and it is number one is depend on the estrogen receptor expression, and some statistic about that. 60 to 70% of breast cancer express estrogen receptor, and they respond well for treatment by estrogen by anti-estrogen therapy. Unfortunately, the estrogen receptor negative usually more aggressive and they do respond to therapy. The second one is HER2 or NEO, which have 20 to 30% of breast cancer have amplification of HER2, and they also more aggressive, higher grade cancer, and they respond to treatment with Herceptin. And this is how HER2 amplification by FISH analysis is happening. This one on the left is the normal. You're supposed to have 2 copies. And here in breast cancer, you can see there is many copies of HER2. The third classification or the third subtype is called human basal-like breast cancer, and they call it basal-like because it expresses basal keratins. It is higher grader, and also it is estrogen receptor negative, progesterone receptor negative, and doesn't harbor amplification of HER2.
Please, Dr. Amal, please.
Yes.
Can you begin the presentation from the start on PowerPoint?
Sure.
We see the conclusion, important findings only. The PowerPoint.
Really?
Yes, that's fine. Thank you.
You want me to start from the beginning?
No. No, continue, Dr. Amal.
Continue? Okay. Is that okay? You can see it now?
Yes.
Okay. So we were saying the third subtype is called human basal-like, and they call it basal-like because it expresses basal keratin. Unfortunately, it is estrogen receptor negative, HER2 negative, and also, or not HER2 negative, it doesn't harbor amplification of HER2. So these account for more than 15% of human breast cancer, and unfortunately, it affects younger women and have overall poor prognosis. So one of the ways to classify breast cancer is to do gene expression profile. And one of these gene expression profile, as you see here, just you are looking for the expression of thousands of genes in different type of cancer and try to make a classification for breast cancer. The green means it is expressed, the red means it doesn't express. And as you see here, with the gene expression profile, also it gives the same classifications that I talked about it. You can see the normal, which is the green here. It's a group of genes together, and the basal-like is alone here. The ERB2 or the HER2, which have amplification of HER2, is grouped together, and the luminal with a different subtype of luminal, which mainly also it is estrogen receptor positive, is also classified as a one group. It was interesting that in when they did the gene expression profile, they found that breast cancers that have BRCA1 mutation also have the same genes like the basal-like breast cancer. So the idea was there must be something in common between BRCA1 and basal-like. And like we try to connect the basal-like breast cancer with one known genes or one known mechanism to try to understand or find a therapy for basal-like, because until now there is no therapy for the basal-like breast cancer. So just a small, in this slide, just something small about what is oncogene, what is tumor suppressor gene.
Can you please make the slides go with your talk, because we have only the first one. Can you move the slides so that we have the slide that you're talking about?
PowerPoint slide.
What's the problem?
The PowerPoint slides, not with your talk.
Not moving.
Not moving with your talk.
How come I have the, I'm sharing.
The screen is on the fourth one, on the fourth slide.
Maybe it have something to do with the internet, because for me, what I'm talking is the ones that I see in front of me. You wanted me to go back?
No. Well, we went to the slides. The fifth or sixth slide.
Move the slides.
Move the slides
Move the slides forward.
with your talk.
Move the slide. Okay. So which slide you are at now?
Fourth one. Fourth. Number 4.
Number 4. So I go back?
Yeah.
No,
Forward.
forwards.
Okay. This one?
No, not.
So it is backward. Let me do something.
It doesn't move with your talk.
Okay.
Okay, that's it's the 9th one. This is number 9.
This one?
This one is number 9.
Oh, okay. So it moves faster that I'm talking?
Slower. I think slower.
Lower? So these are the ones that you have now, the one with the hereditary basal-like and the sporadic?
Yes, that's one. Yes, that one.
That's one. So I have to go back. So it moves faster than I am.
Okay.
Because I was here talking about oncogene. Let's do it that way better.
Okay.
You can, is that better for you?
Okay.
And you usually have difficulty, يعني كل مرة بيبقى في مشكلة في
يعني حضرتك بتبدأي والسلايد لسه زي ما هي في نمبر 4 واي ثينك يعني حضرتك في 7 أو 8 كده يعني.
Okay. طب انتوا شايفين السكرين اللي فيها two cars؟ اوكي. Okay. So دلوقتي we will talk about oncogene and tumor suppressor gene for people who don't know. We usually say your cell is like your car. So either for your car to go forward, either you press on your speed, or if you wanted to stop your car, you press on your brake. So the car usually will go forever if you either press on your speed, if your brake have a problem. So your brake is usually your tumor suppressor gene. So we were talking about BRCA1. So BRCA1 it is abbreviation for breast cancer associated gene 1, which is a tumor suppressor gene required for maintenance of genome integrity. It is important for many pathways like the DNA repair pathway. Women with germline mutation for BRCA1, they have 50 to 80% chance to develop breast cancer in their life and 20 to 40% chance to have ovarian cancer. They develop this germline mutation BRCA1, usually they have a cancer profile as a basal-like, as we said from the gene expression profile. Cells with BRCA1 deficiency, they have a defective in DNA repair pathway. So the idea was, as I told you from the gene expression profile, there must be something, correlation or similarity between the basal-like breast cancer and the BRCA1 breast cancer. So first we classify breast cancer into hereditary or sporadic. Hereditary, which the woman will inherit a mutation from her mom, or hereditary which run in the family. Sporadic, which will happen during her life. So in the hereditary, we know there is a BRCA1 mutations. Usually those people also have a checkpoint mutation P53, and could be the trigger for the cancer and genomic instability. While in the sporadic basal-like, usually BRCA1 is intact, but there is another abnormality in different gene down in the stream, and this is code to cancer and genomic instability. So first, what is the DNA damage response? So usually we expose to different DNA damage agent or reagent during our life. And there is a protein called sensor, which usually sense if there is any damage. And another protein called transducers. And those transducers, they carry the message that there is a damage in our DNA and carry that to another protein called effectors. And those effector protein start either doing a repair or stop the cell cycle or start transcription of certain genes or proteins that is important for repair. Or if the cell fails to repair the damage, it will commit suicide and stimulate a mechanism called apoptosis. As you see here, there is many proteins that is involved in this process. Actually, there are more than this, but this is we can say this is the most common, like ATM, MDC1, 53BB1, BRCA1. Okay. Are we okay up to now?
Yeah, okay.
Okay. So the idea was to create a basal-like breast cancer, which also have BRCA1 deficient, and try to understand what is the correlation between two, what is the benefit of that. So we create a mouse that also have basal-like breast cancers that have target knockout of BRCA1 and P53. So knockout means the tumor was deficient in BRCA1, and usually we do also make it deficient B53 because if B53 is intact, the cell will die. So we usually do combination of BRCA1 B53 in order to keep the cell survive or available. So as the beginning, we wanted to understand what different protein or gene expressed in this cell line. So as you see, the 114, we call it 1142 for the mutant one, which have BRCA1b53 deficiency, and the wild type was 1146. So here we look for the expression of BRCA1, and this is a Western blot. And for people who don't know, Western blot is a technique you do to see if the protein is expressed in this cell line or not. And you can see here, this is how the wild type look like, the band at 250, while in 1142, there is no expression of the protein. And this one for the control to be sure that you loaded the same amount of protein. So one of the genes that we found that is also not expressed in this cell line is called 53 binding protein 1. And you can see here in the mutant cell line, there is no expression of this protein. This is a control, which shows that we loaded the same amount of protein, but this 53 binding protein 1 was not expressed in this cell. This is a different technique to show how the expression is. This technique is called immunostaining, where you use the fluorescence technique to see the expression of the protein. You can see here in the wild type here how the protein is supposed to look like. And you can see in the mutant cell lines, there is no expression at all of this protein. And this was very interesting finding because 53 binding protein 1 is one of the important protein in the repair pathway. So we want to know, first, this is some information about 53 binding protein 1. It hyperphosphorylated response to ionizing radiations and localized to the site of histone H2AX, which is, we consider H2AX as, how to say it, like a trigger for the response, or actually it's a protein that inform and initiate the response to the DNA repair. When we use siRNA to silence 53pp1, the cells show a defect in entering the S phase and also show a defect in DNA repair. So back to know, so what is the mechanism why this cell line is deficient in this protein? So we go back to the old biology class, and we know the DNA gets replicated, then after that transcribed into mRNA, then the mRNA will be translated into a protein. So if we don't have a protein, so this means the problem either in the protein or in the RNA or in the DNA. And we use different methods to try to find out where is the defect itself. So to do that, as I showed you before, we look at the protein by Western blot. There is no protein expression. So we did something called Northern blot for the people who's not familiar with the technique. So Western blot, you look at the protein using antibodies. Northern blot, you look at the mRNA using a probe, which you prepare it using the sequence of these genes. So you can see also, here is the wild type cells. You can see there is expression for the mRNA. In the mutant cells, there is no expression of the mRNA. And this is a control loading, again, to show that we load the same amount, but actually the mutant cell doesn't express the mRNA. So we look at the protein level, going back here. We looked at the protein, we looked at RNA, so there is no protein, no RNA. Now we're looking back to the DNA. So looking at the DNA, we look at the genes. It's a big gene. It is about 6.6 kb. That's the mRNA. And I design different primer to cover the whole mRNA. And you can see here, this is a sequence by sequencing. This is a wild type cells, and this is a mutant cell. And when you follow the sequence, you will see they match exactly until this A. From the G, this one, the sequences start to be different down here. Actually, it was two overlapping sequence. One is similar to the wild type, exactly match, and the other one is not. Okay, so it was like, we try to identify what this sequence is. So this was a cDNA. And before I go to my results, we'll go a little onto the RNA processing. As you know, after the mRNA transcription, there is another processing that must happening to the mRNA, which means splice out the intron. First capping, polyadenylation, which adds a tail to the mRNA, then splicing, which means removing of the introns. Then now we have the, we transport the mRNA after all this editing out to the ribosome or the cytoplasm to go to the ribosome where it is start protein assembly. So again, this is how the gene structure for any gene. Usually you have intronic sequence, you have exons, different exons. At the boundary of the exons, usually there is two nucleotides at the beginning and at the end. Usually have AG at the beginning or GT at the end, and it is usually that way. This is a boundary where the it's called spliceosome mechanism come and cut the intronic and remove the intronic sequence from the gene structure or from the mRNA. Any abnormality in these two nucleotides could lead to either miss one exon, add another exon, or stop the protein at one exons. So it could be missing one exon, adding another exon, or what we have here was something very different. So here we have in these genes, that's exon 22, and this is exon 23. And here was the intronic sequence that is precede exon 23. You can see here it was just one nucleotide difference from C to G and leads to change these two nucleotides from AC to AG. And if you notice here, this is the boundary of the exon 23, that is the AG, that is supposed to be a splice acceptor. And instead of this one, the cell use this one and leads to addition of 7 nucleotides, 1, 2, 3, 4, 5, 6, 7, to the sequence, and addition of 7 nucleotides, which is not a multiplication of 3, will lead to frame shift and stop the expression of this mRNA. So this proves that this one nucleotide difference leads to frame shift in the mRNA, and the frame shift in the mRNA also stimulate another mechanism called decay, where the mRNA will go for destruction and no expression of this mRNA will happen. And this is what we said here. There is no mRNA expression, there is no protein expression of these genes. So we find now the conclusion of what up till now we find that this protein is not expressed in this cell line, and we know the mechanism. Now we want to know what's the clinical benefit of that. So we went back to the gene profile expression for this cell line, in a breast cancer collection. These are clinical data from patient. And we looked to the expression of 53pp1 in different breast cancer subtype. And we find that it is low expressed in all these different types, but mainly in the basal-like that we were talking about before. And this proves that there is a connection between BRCA1 basal-like, and this connection may be it's a 53 binding protein 1. Now, this one was with a gene expression profile, and here we're using immunostaining to look for the expression of this protein in a collection from a patient. We have over 400 breast cancer specimen in tissue microarray, did immunohistochemistry. So you can see this is a positive, which expresses the protein. This is a negative. And our data shows that 14.5% of all tumor samples were 53 binding protein 1 negative. 90% of this was mainly in the triple negative that, or the basal-like that we talked about it at the beginning. So again, these data confirm that there is a connection between the basal-like, the BRCA1, and the connection is 53 binding protein 1. Now we find also by looking to a data, it's a collection of information, clinical information about patients. And we correlate the expression of this protein and the metastatic free survival for those collections. And we find out that women with expression of 53 binding protein 1, they tended to survive longer or do much better than women with lost this expression. And we came to the questions. Again, all the work is to try to find a connection between BRCA1 and basal-like, because you usually know that BRCA1, we have a treatment for BRCA1 now, or there is a trial for treatment for BRCA1 with a PARP inhibitor. And the idea was, can we use the same treatment for the basal-like also, because of the connection between BRCA1 and basal-like? So now we came to our proposal at the beginning, or our hypothesis. We now conclude after that from all the work that we did, yes, there is a connection between basal-like and BRCA1, but we need to be clear about it. So we said in the hereditary basal-like, BRCA1 is deficient, and we find through the cell line that also 53 binding protein 1 is deficient. So there is a connection here between BRCA1 and 53 binding protein 1. In the sporadic, which is BRCA1 is intact, but we find through the collections that we have that 53 binding protein 1 is deficient in the sporadic one. So from this, we know now it's 53 was it is present, it's deficient in both hereditary and sporadic. And maybe this is the reason why both, they have similarity. And again, we conclude that in another work. And this is, again, we used some, it was a bigger, the other one was 400 cases. Now we use 1,043 cases, and we still see the same, that all breast cancer have deficiency of 53 binding protein 1, but some of them also have BRCA1 deficiency. Going to the therapeutic part, so we use the same cells that we work on it at the beginning, that is BRCA1 deficient, B53 deficient, and we try to use knockdown that B53 binding protein 1 also, and we can notice that here cells that knock out both BRCA1, B53, and 53 binding protein 1, they survive longer than the one that is BRCA1 deficient. And this means that this cell line will, the treatment with cisplatin and mitomycin may be not good when you lose 53 binding protein 1. And this was, was good actually, not because there were some reports coming out after the first trial with the PARP inhibitor, and we figured out that certain women with a BRCA1 deficiency, after they received the treatment for PARP inhibitors, they started to have resistance later on for it. And we felt like our work showed that 53 binding protein 1 status, it changes the response to the treatment. So we tried to find out if also this could be related to the loss of our protein, which is 53 binding protein 1. And yes, we did that. We treat the tumors with PARP inhibitor, and at the beginning you see there is complete response. Then later on, some tumor, they are not responding anymore for the treatment, and this is the immunostaining for 53 binding protein 1, and 2 of the tumors that develop resistance, we find out later that they also lose 53 binding protein 1. So it seemed like the cell tried to overcome the treatment and try to lose 53 binding protein 1 and overcome in order to stay and survive the treatment. So that is the end. I hope I was not that fast for you. So the findings that we found this protein, which is called 53 binding protein 1, expression is reduced in subset of sporadic and BRCA1 triple negative breast cancer. And we proved that in a cell line, and we proved that in breast cancer tissue. And we also proved that loss of this protein may tolerate the BRCA1 loss and alleviate the DNA repair defects that associated with the BRCA1 loss. And this protein also have significant role in development to resistant to PARP inhibitor and cisplatin. And actually now in the era of personalized medicine, they started to do gene profiling for a woman who is receiving PARP inhibitor or cisplatin. And the recommendation now, it is not yet, it will be that to look at the status of her gene profile, if there is a 53 BB1 intact, this means she will not benefit from the treatment, and it's better to avoid giving her this treatment because she will get the side effect with no benefit from the treatment. And I think that is my last slide. I hope you like it, and I hope I was not that fast. This is my first lecture to Egypt, and I'm happy to hear from you. And if you have any question, any suggestions, I'm open for any further discussion.
Okay, thank you.
You're welcome.
Thank you, Dr. Amal, for your presentation. I just wanted to know if this 53 binding protein is available for to be used as immunohistochemical staining now?
Yeah, it's available. The protein is commercially available, yes.
Okay. And so if we find that the staining is positive, so we can use the BRCA1 treatment in these cases?
If it is positive, you cannot, yeah, you can use BRCA1 treatment for these patients.
And it is, this 53 is different from the P53 that we know.
Yes, actually, because of the beginning of our lecture, so to be honest with you, I missed some stuff. I was preparing a lot to say, but I supposed to start with you 1 hour earlier than, but because of the time difference between Egypt and here, the time was not correct. Then when we start late because of the connection. So, but B53, which you call it the guide or
the guardian of the
genome. We use it actually as a bait, which means like Sinara to catch different other. They use it in a yeast experiment to try to find other proteins that work with P53 and help P53 in binding to the DNA. And they find 2 proteins. One is called 53 binding protein 1, and the other one is called 53 binding protein 2. And this happened in 1996. And since that time, more attention was to 53 binding protein 1 because it is involved in many DNA repair. And it's considered like the second line of protection for the cell in DNA damage response.
Okay. So we start with P53, the regular one, and if it is positive, we can use the P53 binding protein 1.
Yeah, usually what I know that they are doing right now in the clinical research, they're doing staining for 53 binding protein 1 because usually most of cancer they have a mutation P53. Okay. So what we're talking about here, they look to BRCA1. If BRCA1 is deficient, they do 53 binding protein 1. If it is positive, this means the patient can benefit from the therapy. If it is negative, this means the patient will not benefit from the therapy.
Thank you very much. Thank you very much.
You're welcome.
Okay, we hope next time the connection will be better and we can start at the proper time, and we'll have more time to hear from you.
Thank you. I have a question to you. I know I present mostly in English. Do you prefer that, or next time you want me to speak some Arabic? أنا بتكلم عربي كويس جدا يعني. But I really didn't know how to do this my first time, so I would like to hear from you what you think.
Well, English is okay. If you can put some Arabic, they will be happier than that.
Okay.
But English is okay for all of us.
That's good. أنا حبيت أسمع منكم لأن دي أول مرة. I know maybe this not really interested for many people. المرة الجاية إن شاء الله I'm planning to talk about the board of medical genetics للناس اللي هم إن شاء الله interested إن هم يجوا أمريكا ويتعلموا أو ياخدوا board. هتبقى I will share you with you إزاي to apply requirement. هي requirement بتاعته مختلفة شوية برضو عن other board لأنه في clinical وفي laboratory subspecialty فيه، فالrequirement للclinical مختلفة عن اللابوراتوري. الأوبشن بتاعته كويسة جدا لناس كتير اللي هم مش عايزين يعني اللي هم they prefer the laboratory side، اللي هم بيحبوا أكتر المجال بتاع lab، خصوصا إنه معظمكم على حسب ما أنا فهمت this is the pathology department.
Yes.
Yeah. The Board of Medical Genetics, it can be subspecialty of pathology, and you don't have to go through the headache بتاع other board. فإن شاء الله دي تبقى المرة الجاية بإذن الله.
إن شاء الله.
إن شاء الله.
Thank you very much.
You're welcome. Thank you very much for all of you.
Thank you.
سلام.
عليكم السلام. باي باي.
Bye-bye.